Okay, students, so we are back with the second part of biotechnology principle and processes. In this part, we are going to discuss about remaining tools of biotech, that is your enzymes, foreign DNA, and host cell. Okay, so let's get started with this. So first we are now having, in the previous lecture we studied about vectors, the vehicle DNA, okay, that can be your BAC, bacterial artificial chromosome, your YAC, yeast artificial chromosome, plasmids, phages, all these are different kinds of vector which we use. Vectors are those things in which we glued our gene of interest and set inside the host cell to multiply to do replication. In that lecture we also studied the features of your plasmid, ideal plasmid, the characteristics of ideal plasmid, that we studied that it should have that rope, okay, then we have it should have origin of replication, high replication rate. Fourth, we studied that it should have selectable marker, okay, and then we studied about different items, different plasmid, and the most usual and most useful and the most usable plasmid in biotech, that is your PBr322. Okay, so these we studied in the previous lecture. Now we are going to discuss about the enzymes, that cleaving enzyme, different types of all type of enzyme we are going to discuss in this part. Okay, so recall the vector as we studied, your BSC vector DNA fragment, that is your gene of interest that get attached and we get a recombinant DNA. Okay, vector act as a carrier or vehicle for the DNA fragment which transferring into the host cell. These all things we studied in the previous lecture. Okay, now first, starting from this enzyme part, we have to discuss some basic knowledge. Okay. For example, we have to know about bacteriophage, how that bacteriophage that infects bacteria, and in the infection, what that bacteria do against that bacteriophage genome. Okay. So first, bacteriophage or phages are the virus that infect bacteria and grow on them. Many bacteria are susceptible to these viruses. Okay. So many bacteria are susceptible to these viruses. Okay, these viruses attack to the surface of bacterial cell, inject their genetic material, and use bacterial enzyme to multiply while destroying the bacterial cell. That's how they will do infect your bacteria. Same mechanism that HIV done with our helper T cells and macrophages. These bacteriophages injects their genetic material inside the genome of bacteria and multiply inside that bacterial genome. Okay, so this is your bacteriophage, it contain its genetic material. Okay, so it attached to the surface of the bacteria, cell wall of the bacteria, and injects its genetic material inside that genetic material it start doing transcription, translation, and produce its own copies of its genetic material, and after some time it have the millions of copy inside the bacteria. That's how your bacteriophage do division and reproduce inside the body of bacteria. Okay, so now after isolation of gene of interest, it needs to be inserted into the host cell to produce the desirable protein that we studied in the previous part. Okay, however, gene of interest cannot be inserted directly into the host, and it required a vector to take it into the host. Okay, you cannot directly insert that gene of interest inside the bacteria inside the host. Okay, again I am saying you cannot directly insert the gene of interest directly inside the host. Okay, you need some vector, okay, that we studied in previous part of the lecture. Okay, so plasmids are the most commonly used vector. Okay, your plasmid are the main and the most common used vector in biotechnology that we all studied in the previous part. Plasmid are autonomous replicating extrachromosomal circular DNA found in the bacteria, that also studied in the previous part, that bacteria are extrachromosomal DNA which replicate inside the cell of bacteria. Okay, that we studied in previous part, the first part of biotechnology. Okay, to insert gene of interest, the circular plasmid need to be cut open, which can be done by endonucleases. So in this part we are going to study about this endonuclease. Okay, again I am saying these all things we studied previously. Now in this part we are going to study about endonucleases. Okay, endonucleases and different enzymes which we use in biotech. We are going to use. We are going to study that all the things. Okay, as you know, enzyme, the second tool of biotechnology. In this lecture, we are going to discuss about the second tool of biotechnology, that is enzyme. Okay, so let's get started now. So enzymes, a number of specific kind of enzymes are employed in genetic engineering. These include lysing enzyme, cleaving enzyme, synthesizing enzyme, and joining enzyme. These are the categories of enzymes which we use in biotech. So first we have lysing enzymes. It's a word, its name, the word saying its meaning. Lysing means it is to degrade, to lyse something. Okay. So these enzymes are used for opening the cell to get DNA for genetic experiments. Okay. So for different types of cells, we have different types of lysing enzyme. For bacterial cell, we have that lysozyme. For plant cell, we have cellulose, pectase, okay, all these. Then for fungal cell, we have chitinase, okay. But in animal cell, we do not require any lysing enzyme because in animal cell, you do not found any cell wall. Okay, so these are the enzymes which will use to degrade the cell wall of that host cell. Okay, so bacterial cell is commonly digested with the help of lysosome. Plant cell is commonly digested with the help of cellulose and pectinase. Fungal cell is commonly digested with the help of chitinase. Okay, next we have cleaving enzyme, and that is the most important type of enzyme in biotechnology, in genetic engineering. Okay, that is exonuclease category and endonuclease category. Okay, these enzymes are very important tool in biotech. Again I am saying, okay, so these enzymes are used to cleave DNA molecule. Cleaving enzymes are of two types: exonuclease category and endonuclease. Exonuclease are those enzymes which cut DNA from outside, and endonuclease are those the enzymes or those category of enzymes which cut DNA from inside. Okay, so exonuclease remove nucleotide from the ends of the DNA from the outside, and your endonuclease may cut at specific position within the DNA. Example, restriction endonucleases. Okay, so again I am saying two types of that cleaving enzymes are there: your restriction endonucleases or endonuclease categories, and exonuclease category. Exonuclease cut from outside, it cut nucleotides from the outside of the DNA, while your endonucleases cut the DNA from the middle or a specific side, but always inside the middle of the DNA. Okay. Next category we have synthesizing enzymes. Okay, these enzymes produce something. They are used to synthesize something. Okay, so these enzymes are used to synthesize new strands of DNA complementary to existing DNA or RNA template. They are of two types: reverse transcriptase and DNA polymerase. Okay, these are to used to synthesize DNA and RNA strands. Okay, so we have two types: reverse transcriptase and RNA polymerase. So reverse transcriptase produce DNA from RNA. Again, I am saying reverse transcriptase produce DNA from RNA. Okay, it helps in synthesis of complementary DNA strand on RNA template. Okay, then we have DNA polymerase, help in the synthesis of complementary DNA strand on DNA template. Okay, DNA polymerase produces DNA from DNA. Our reverse transcript produces DNA from RNA. From a primitive thing, you are producing an advanced thing. That's why it is called reverse transcriptase. Normal in molecular basis of inheritance, you studied transcription. Means you have DNA, and from that DNA you are forming mRNA. But in reverse transcriptase, in reverse transcription process, what happens? You have RNA and you produce DNA from that RNA. That process is called reverse transcription. An enzyme which is used to do this is called reverse transcriptase, a category belong to a category of synthesizing enzyme. And the last category is joining enzyme. They are molecular glue. Okay, they joint in DNA particles, they joint DNA strands. Okay, so these enzymes help in joining the DNA fragment. For example, DNA ligase is used to join DNA fragment. Joining enzymes are therefore called molecular glue. Okay, so you are joining enzyme category in which we have DNA ligase, which is a type of your ligating enzyme and also called as molecular glue. Your exonuclease and endonuclease is that cleaving enzyme. They are called molecular scissors. And this joining enzyme DNA ligase is called molecular glue. Okay. So this is we studied till nuclease are the enzyme that cleave nucleic acid, DNA and RNA by breaking down the phosphodiester bond. Nuclease, we have based on the region of action, we have endonucleases and exonucleases. Okay, we have also one category based on the substrate on it is acting. Okay, on the basis of that we have RNAs and DNAs. Okay, that is the type of nucleus. RNAs for RNA and DNAs for DNA. Okay, but this category is based on region of action. So endonuclease may cut within the DNA molecule. Okay, you can see here it will cutting from this region only. Okay, it is cutting from this point only in the middle of the DNA. It is cutting, not from outside. Okay, then it is your endonuclease. But in this exonuclease may cut at the end of the DNA molecule. You are seeing that it cutting outside, not in the middle, not in the some center part of the DNA. It is cutting only outside. Okay, it is cutting only outside. Then it is your exonuclease. And when an enzyme cutting from the middle or a specific region inside the DNA, then it is your endonuclease type of that cleaving enzyme. Okay. Now, अब ये restriction enzyme की discovery कैसे हुई? How your restriction enzyme discovered? Okay. So restriction endonuclease are type of endonuclease found in certain bacteria that cut DNA molecule at a near specific nucleotide sequences. Okay, it was discovered as a part of restriction modification system in bacteria. Okay, बैक्टीरिया अपने आप को बचाने के लिए वायरस से एक सिस्टम चलाता है. उस सिस्टम को बोलते हैं रेस्ट्रिक्शन मॉडिफिकेशन सिस्टम. Okay, your bacteria wants to save itself from viral attack, that bacteriophageal attack. So it will do what? It will start a mechanism which is called restriction modification system, in which it initiates that restriction enzyme which cut the genetic material of your bacteriophage. And if that genetic material is degraded, then automatically your bacteriophage cannot divide inside that bacterial cell. Okay, so by which is used by bacteria to protect its own genome and destroy the foreign DNA, example bacteriophage DNA. So restriction modification system have two, restriction enzyme and modification enzyme. Two enzymes come under restriction modification system. One is restriction enzyme, another one is modification enzyme. Restriction enzyme cuts your viral DNA, viral genome, while modification enzymes save the genome of bacteria from this restriction enzymes. Okay, let's see that thing in detail. Okay, see, this is bacteriophage. Okay, this is bacteriophage. Okay, now this bacteriophage contains DNA. Now it makes its DNA enter inside the bacterial cell. Okay, now it will start to divide. But at that time when it get enter inside the bacterial cell, your bacterial genome secrete two enzymes. In the protection of bacterial cell, one is your that is restriction enzyme, and second one is your that compressive enzyme or that maintaining enzyme. Okay, so this restriction enzyme go and cut your DNA of bacteria, the genetic material of bacteria. It is cutted by your restriction enzyme. Okay, देखो कटा हुआ है काटी, that scissor is made there. Okay, so it recognize and cut up the foreign bacteriophage DNA. Okay, then here on that your मतलब ये जो चीज है ये जाके अपने डीएनए को ना काट दे, उसके लिए आता है मॉडिफिकेशन एंजाइम. एंड व्हाट मॉडिफिकेशन एंजाइम डू? इट एड्स मिथाइल ग्रुप टू योर बैक्टीरियल जीनोम. सिंपल है, सिंपल. देखो क्या है, बैक्टीरिया के अंदर बैक्टीरियोफेज ने अपना जेनेटिक मटेरियल डाला. Okay, now that genetic material have power to divide inside the bacterial genome, inside the bacterial cell. Okay, now your bacterial cell releases two types of enzyme. One is restriction enzyme and second one is modification enzyme. restriction enzyme act on the DNA, on the genetic material of bacteriophage and make it into a fragment, while modification enzyme works on the genome of its own bacteria खुद के genome के ऊपर काम करेगा और उस पर मिथाइल ग्रुप चिपका देगा, and that methyl group prevents the action of this restriction enzyme on its own bacterial genome. Okay, we can draw this thing here. By drawing you can see here. Okay, देखो देखो ये है बैक्टीरियोफेज. ओके, ये गया और बैठा तुम्हारे बैक्टीरिया के ऊपर. ओके, it releases its genetic material inside this. Okay, now this is the genome of bacteria. Okay, now from that genome two enzymes will come. Okay, one is your modification, another one is restriction. Okay, now this restriction enzyme works on this bacterial DNA. Okay, sorry, bacteriophagal DNA. Okay, now this modification enzyme work on your bacterial genome and aid methyl group to your bacterial genome. Okay, now that bacterial genome become methylated. Now this restriction enzyme does not come and cut its own DNA because of adding of that methyl group. That simple thing it is. Okay, so that thing is written here. It recognize and cut up the foreign bacteriophage DNA, that is your restriction enzyme, and modification enzyme, it recognize and modified the bacterial DNA by adding methyl group to protect it from the DNA degrading activity by its own restriction enzyme. वो खुद का रिस्ट्रिक्शन एंजाइम आके खुद को ना मार दे, उसके लिए बैक्टीरियल डीएनए, बैक्टीरियल जीनोम दैट मॉडिफिकेशन एंजाइम निकालता है और अपने डीएनए को मॉडिफाइड कर देता है बाय एडिंग मिथाइल ग्रुप इनसाइड द डीएनए. ओके, so did you know restriction modification system discovered in E. coli? Okay, this restriction modification system is first discovered in E. coli. Okay, this is your E. coli. Okay, second thing, mode of action. Now how your restriction enzyme do the work, how this enzyme works on the DNA, let me see that thing. Okay, so restriction endonuclease scan the DNA for certain specific sequence for nucleotide. Okay, ये जाता है और तुम्हारे डीएनए को रीड करता है. ओके, इट विल गो एंड रीड योर डीएनए एंड फ्रॉम अ सर्टेन पॉइंट, फ्रॉम अ स्पेसिफिक पॉइंट, इट कट डाउन द डीएनए. ओके, the specific sequence which are recognized by restriction enzymes are called recognition sequence or recognition sites. Okay, जो the sequence which is readed by your endonucleases are called recognition sites or recognition sequences. Okay, and that sequence is cut down by your endonucleases. Okay, recognition site helps the restriction endonuclease to identify the cut site on the DNA. Okay, that recognition site identify and help your endonucleases to cut the DNA on a perfect way, on a specific way. वरना ये तो कटिंग चीज है, कैंची है, चलेगी तो सबको काटती चली जाएगी. तो क्या करती है ये, स्पेसिफिक साइट्स को रीड करती है एंड फ्रॉम दैट स्पेसिफिक साइट इट कट्स द डीएनए. Okay, so did you know more than 900 restriction enzyme has been isolated from over 230 strains of bacteria? Till now you have 900, more than 900 restriction enzymes which has been covered, which has been secreted, which has been discovered, that isolated from 230 strains of bacteria. Okay, so that is a general knowledge thing. So now अब तुम्हारा जो रेस्ट्रिक्शन एंडोन्यूक्लियसेस होते हैं वो एक पैलिंड्रोमिक सीक्वेंस पे चलते हैं. ओके, इट कट्स योर डीएनए इन द फॉर्म ऑफ पैलिंड्रोमिक सीक्वेंस. ओके, So in English, palindrome refers to the word which reads the same from left to the right. Okay. Example, Malayalam. Okay. Level, rotator. Okay, you will see Malayam. Up from backward, back side se padho. Malayam. Okay, that is called palindromic sequence. स्टार्टिंग वही है, एंड भी वही है, दोनों एक दूसरे से सेम है. ओके. लेवल LEVEL, सेम सेम. रोटेटर ROTATOR. ओके. आगे से पढ़ो या पीछे से पढ़ो, सेम वर्ड. Okay, when you read from starting or you start to read from the end of the word, that are same, that thing is called palindromic words. Okay, same thing your restriction enzyme do, same type of cutting your restriction enzyme do, that is called palindromic sequence cutting. Okay, so some restriction enzyme have recognizing site that reads the same on the both strand, that when the orientation of reading is kept the same. Example 5 to 3. Okay, these sequences of bases pair in the DNA are known as palindromic sequences. Example, EcoR1 cut the DNA between the base G and E only when the sequence GATTC is present in the DNA. Okay, now you have a sequence that is GAATCC. Okay, so what will be its opposite? It will be CTTAAG. अब देखो, पढ़ो ऊपर से. GAATTC उल्टा करो, CTTAAG. Okay, again GAATTC, that is the sequence your EcoR1 will cut. Okay, now when you do make a palindromic sequence on the opposite, if it is 5 to 3, then it will be 3 to 5. Okay, so it will be CTTAAG, opposite, same. Okay, on opposite strand the same sequence is there, but from you have to read from backward. अगर यहाँ से G से C पढ़ रहे हो तो उसको उल्टा पढ़ो, CTTAAG, तो नीचे वही सीक्वेंस तुम्हें दिखेगा. दैट इज पैलिंड्रोमिक सीक्वेंस कटिंग. ओके, देयर इज वन मोर इमेज. ओके, AAGCCTT. ओके, उल्टा कर दो, TTCGAA पढ़ो, so TTCGGA. ओके, फ्रॉम फिफ्थ टू थर्ड डायरेक्शन you have AAGCTT. okay, and when you revert that thing, okay, from 5 to 3 direction, then it will come TTCGGA. कुछ नहीं है, सेम है. ये डीएनए तुम्हें पता है एंटी पैरेलल होता है. ओके, डीएनए इज एंटी पैरेलल. ओके, इट विल मींस ऑफ द अपर स्ट्रैंड इज 5 to 3, देन लोअर स्ट्रैंड विल बी 3 to 5. ओके, तो अगर ये 5 से 3 डायरेक्शन में AAGCTT चल रहा है, तो सेम चीज उल्टे उसमें चलेगी 3 to 5 में. ओके, TTCGAA AAGCTT, that thing is called palindromic sequencing. Okay. Next we have action of restriction enzyme. Now how your restriction enzyme acts. Okay, restriction enzyme cut DNA into fragment of the size suitable of cloning. The restriction enzyme produce two types of cut. Your restriction enzyme cuts DNA in two forms. One, blunt end, and second one is sticky end. Blunt end means it cut from the middle. Okay, middle में से काट दिया, दो हिस्सों में बट गया DNA. It will cut from the middle and separate your DNA into two parts. And sticky in the zigzag manner. Okay, see, this is blunt. Okay, बीच में से काट दिए, दो हिस्से में कन्वर्ट हो गया, सिंपल. ओके, ये देखो, ये है तुम्हारे पास एक डीएनए का स्ट्रैंड, 3 to 5, 5 to 3. इसको तुमने बीच में से काट दिया. ओके, 5 to 3, 3 to 5, 5 to 3, 3 to 5, two different strands are there. That thing is called blunt end, and it is produced by some that restriction enzymes. Okay, this is blunt. Okay, now this is your DNA sequence. Okay, and now your restriction and that restriction endonuclease creates sticky end. So it will do what? It will not cut like this, it will cut like this. See, it will cut like this. This is one strand, this is one strand, and they can be attached. Okay, this is your sticky end. Okay, this is your sticky end. Okay, and when you talk about like this is your blunt end. Okay, this is blunt end. This is the blunt end cutting by your restriction endonuclease, and this is your sticky end cutting by the help of restriction endonucleases. Okay, so see in this image you can see here it will cut from 5 to 3 direction. Okay, in the same manner, okay, and creates a sticky end. Okay, this is overhanging, this one and this one. Okay, in this you can see this one and this one, this and this one, these both are overhangings, they get attached from each other. Again the hydrogen bond will form again here. Okay, hydrogen bonds and the phosphodiester bond will form again. Okay, now so these are different your restriction enzyme and their cutting sequences. Okay, so your balm H1 it will mix sticky ends. Okay, then your eco R1, okay, that also forms sticky end. Then your hind 3 it also forms sticky end. Then PSTL1 it is also forms sticky end. Then you have PUV 2, okay, it forms blunt end. Then you have SAM1, it also forms blunt end. Okay, and these all are different restriction endonucleases. BAM H1, ECO R1, HIND 3, PSTL1, these form sticky end, while your PVU2 and SMA1 they form blunt ends. Okay, and these are the restriction, the recognition sites. Okay, now we have nomenclature. Okay, अभी तुमने पढ़ा ये सब इको R1, BAM H1, HIND 3, PST1, ये नाम आए कहाँ से? How you will give the name to these things? How can you give an enzyme a name like this? So next we are going to study that thing, that is nomenclature of enzymes. Okay, so restriction endonucleases are named according to the organism in which they are discovered using system of letters and number. The nomenclature follows certain rules. Okay, so the first part, let's see. जैसे कि ये है तुम्हारे पास इको R1. Okay, this is your restriction enzyme eco R1. Okay, in which this part, these both, the first letter and the second letter. Okay, the first letter and second letter is your species name and genus name. Okay, the first one is your Escherichia. That is the genus name. Okay, second letter is your species name. Okay, the third letter belongs to the strain. This one is your strain. Okay, and the fourth one is your discovery number, and it is always in Roman number. This is your discovery number, discovery sequence. Okay, again I am saying the first letter is the genus of the organism, the bacteria where you discovered that restriction endonuclease. The second is your species. Okay, the second word represent the species of that organism. Third is your strain of that organism, the bacterium from which you are taking out that restriction endonuclease. Its the strain is given by that number third, represented by the third digit. Okay, that of endonucleases. Then the fourth one, the fourth letter is your number of discovery. At what number it is discovered? If it is eco R1, means it is the first. Okay, then we have eco R2, second bar discovered hua. Eco R3, it will discovered on third time. Then eco R4, fourth time. Eco R5, that is fifth time. Okay, so that is how you can name our restriction endonucleases, or restriction enzymes. Okay, that is same thing written here. Okay, the first part, first letter of the genus name. The second part, first two letters of the species name. Then the third part, first letter of strain. Then fourth part, order of discovery in Roman number. Okay, that I explained now. That example is here. The first part, first letter Escherichia coli ka E. Then the second part, the species two letters co, that is coli. Okay, then the third part strain, that is R strain. Okay, and the fourth part is the order of discovery in Roman number first, that is 1. Restriction enzyme to be discovered in E. coli. Okay, so that's about the restriction nomenclature of restriction enzyme. Okay, same thing is written here. Okay, in form of flow chart. Okay, now we have to insert that genetically your GOI, okay, that gene of interest you have to insert inside the plasmid. How can you insert? By the help of restriction enzyme. Now, how this restriction enzyme cut that thing into insert your GOI? Okay, let's see that thing. Okay, so this is your eco R1 restriction recognition site. Okay, this is having sequence GAATTC TCTTAAG. Okay, see. So this is your eco R restriction site recognition site. Okay, and this having the sequence GAATTC CTTAAG. Okay, now plasmid DNA with restriction site. Okay, now you have to insert desirable DNA, your gene of interest. So how can you cut? Two things are there. First, restriction endonuclease cut that DNA, then ligase joined that DNA. Simple. So gene of interest with flanking sequence after PCR flanking sequence have the suitable restriction sites. Okay, so this is your GATCC, GATTC, or in this middle part, this pink part is your GOI, gene of interest that you inserted inside the genome of that the plasmid of that bacterium. Okay, so a suitable plasmic vector is required for carrying the GOI into the host. A plasmid has several restriction sites which can be exploited to insert the GOI. In the GOI is required to have suitable restriction site in its flanking sequences. At a suitable restriction enzyme is required which can produce the same sticky ends in the plasmid and GOI. Okay, if that site having eco R1 recognition site, then you have to take eco R1 restriction enzyme. It having the sites for HIND3, then you have to take the restriction enzyme HIND3. If at having the that recognition site of SAL1, then you have to take restriction enzyme SAL1. Okay, ligase is required to make the phosphodiester bond between the DNA fragment to join plasmid and GOI together. That is the simple work of your DNA ligase, that it will join this thing by the help of phosphodiester bond between the DNA of plasmid and that GOI, your gene of interest. Okay. So see, this is the how we generate sticky end in the plasmid at GOI, okay, and GOI, okay, generation of sticky end. Okay, so first digestion with suitable restriction enzyme. Okay, so this is your eco R1 site. Okay, now you have GAATCC sequence and CTCAGG, that is reading sequence of eco R1. Okay, now your eco R1 will come, and what your eco R1 forms, it forms sticky end. So will it cut down your DNA like this. Okay, like this. It will cut your DNA like this. Okay, now it's plasmid with sticky end. Your plasmid have a sticky end. Now this area, should be filled, and it is filled by your gene of interest. Okay, see, digestion of same restriction endonuclease. Now you have that gene of interest having same recognition site for eco R1, and it will cut your DNA like this. Okay, now you have what? GAATTCC. Now this G is separated from this AATTC, and now here you have ATTC, and this ATTC will bind with this. Okay, and this CTTA, that your G, that this part of G, it will come and bind here. Okay, that's how simply your matching pairing of that for GOI and plasmid take place. Okay, in the next slide you will see more clearly. Okay, see, Now your plasmid with sticky end and the gene of interest with sticky end, you will going to combine it. So see this green part, okay. Complementary base pairing of sticky end. So this one is your GOI. Okay, ये वाला part, this one. Okay, and this below one from here to here, it is your this part. Okay, then this one, this part is your this one. Okay, and this part is your this. Okay, so that's how compare that complementary base pairing done in your that between that plasmid and gene of interest desirable gene. Okay. So ligase enzyme link the 5,3 phosphate and 3-OH2 fragment by making a phosphodiester bond between them. This process is called ligation. Now you have this thing, all the thing. Now your ligase will come and attach this thing, and you created a recombinant DNA in which you have the gene of desire, and this is the part of your plasmid. Okay. After ligation, the process of insertion of gene of interest into the plasmid completed, and hence recombinant DNA is formed. In the next step, the recombinant plasmid DNA inserted inside the host cell. ओके फिर उसको होस्ट सेल के अंदर डाल देते हैं और होस्ट सेल के अंदर वो रेप्लिकेट होता है. ओके दैट रिकॉम्बिनेंट डीएनए इंसर्टेड इनसाइड द होस्ट सेल, एंड इन होस्ट सेल इट स्टार्ट टू डू replication. Okay, so now we are going to with the summary of the chapter which we done. Okay, so plasmid are the autonomous replicating extra chromosomal circular DNA found in the bacteria. To insert gene of interest, the circular plasmid need to be cut and opened with the help of endonucleases or exonucleases. Okay, then we studied about nucleases. Nucleases are the enzyme that cleave the nucleic acid, DNA, RNA by breaking down the phosphodiester bond. Okay, your nucleases cut the phosphodiester bond which is present inside the plasmid. Okay, then you have restriction endonuclease. Okay, restriction endonuclease are the type of endonucleases which found in certain bacteria that cut a DNA from specific sites and create two types of end. First one is your sticky end and second one is your blunt end. Blunt end means like this. Okay, this is your blunt. Okay, straight blunt. And then second one is your sticky one, that is your sticky one. Okay, complementary base pairing take place in your sticky ends. Okay, then we studied that nomenclature. Okay, nomenclature, then how can we give restriction endonucleases their name? So for example, we have eco R1 in which that E letter belongs to the genus of the bacterium Escherichia coli. Escherichia, E equals to Escherichia. Then we have co, that is coli. Okay, then we have E. coli, eco R1, that R is strain and 1 is the number of discovery. Okay, so the first letter of the genus, then first two letter of the species, and third part of that is strain, and fourth part is the order of sequence. Okay, now in this I am putting that your eco R1 by giving again the example. Okay, we have enzyme eco यार eco R1. Okay, so this E is the first part of the genus Escherichia. Next, co is the second part, that is coli. Okay, then R is the third part, strain, RY strain. And the fourth part is your number of discovery. Okay, it can be one Roman number, then two Roman number. It is always written in the number of discovery. The order of discovery is always written in Roman number. Okay, so that is how you give name to a certain restriction endonucleases. Okay, then we studied how that all this thing take place, how your endonucleases cut the plasmid and your gene of interest. Okay, so it will cut from that palindromic sequences complementary base pairing type. Okay, so it will recognize the site. Okay, first your eco R1 go and search the plasmid where is my site of cutting. The eco R1 recognition site where that eco R1 endonuclease will go and cut the site, and it will find that site and produces sticky end. Same thing this eco R1 do on your gene of interest and cut that gene of interest in the form of sticky end. Okay, with sticky ends. Now you will do what? Base pairing. Okay, complementary base pairing. G के आगे C, T के आगे A, complementary base pairing and form that particular DNA, that is your recombinant DNA. Okay, so this is the gap which is filled with the help of ligase. Ligase do what? It joined the phosphodiester bond and you get a recombinant DNA which is ready to introduce inside the host cell where it will do replication. Okay. Next, this is the second that we studied, the second tool of biotechnology. Now that part is third tool. Okay, the third tool of biotechnology is desirable DNA, alien DNA, foreign DNA और पैसेंजर DNA. ये एक ही चीज के चार नाम है. ओके, जैसे शिव जी का और नाम होता है, जैसे शंभू, भोलेनाथ, ओके, महाशिव, ओके, शिव जी, ओके, पार्वती that husband of Parvati Devi. Okay, so these all are different names of Shiv Jis. Just like that, your foreign DNA having many names. Desirable DNA, alien DNA, foreign DNA, passenger DNA, gene of interest. Okay, these all are different name of same thing. Okay, so it is the DNA which is transferred from one organism to another by the combining it with the vehicle DNA, that is your vector. Okay, the passenger DNA can be complementary and synthetic. Complementary means it is synthesized on mRNA template with the help of reverse transcriptase. Okay, complementary DNA is always produced with the help of reverse transcriptase, while your synthetic DNA is produced with the help of DNA polymerase. Okay, so it is a synthesized on mRNA template with the help of reverse transcriptase and necessary nucleotide, which called cDNA or complementary DNA is formed through reverse transcription, is shortened than the actual or in vivo gene because of absence of intron and non-coding regions. Okay, non-coding regions and your introns are absent in cDNA, complementary DNA. Second is your synthetic DNA. As DNA, it is artificially synthesized with the help of DNA polymerase. Okay, and the first synthetic DNA is produced by Kornberg in 1961 and Khurana in 1968 synthesized first artificial gene without a template. Without a template, a gene is produced with the help of Khurana in 1968. Okay, and at last the fourth tool of the biotechnology is host cell. This cell is used for DNA cloning. Host cell may be plant cell, animal cell, bacterial cell and fungal cell. Okay, so for today that's all. In the next lecture we are going to study about that mechanism, the processes of biotechnology. Okay, thank you for watching. Like, share, subscribe the video.